means that double-stranded DNA molecules present in the cell of organism are the repository of genetic information. A eukaryotic cell has more than one DNA molecule in its nucleus. Each of these DNA molecules carries thousands of genes that determine an organism's inherited rates. This structure is known as chromosome. In Greek, chroma means color and soma means body. Chromosome can be defined as trait structure in eukaryotic cells nucleus that carry genetic information in the form of gene. The complex of DNA and its associated protein is called chromatin. It is the material that makes up the eukaryotic chromosome. Each single copy of replicated chromosome is called as chromatin, while chromatin states are bound to each other at centromere are known as sister chromatin. Homolog chromosome has corresponding DNA sequence and come from separate parents. One homolog comes from the mother and the other comes from the father. What is tetrad? Tetrad is two pairs of homolog chromosomes next to each other.
A centromere is a constructed region of DNA or a structure in a chromosome which plays an essential role in segregation of chromosomes during the process of the cell cycle in eukaryotic cells. Centromere can be defined as the compressed region or a part of elongated chromosomes. It is the specialized DNA sequence in the chromosome that links or holds together the pair of sister chromatids. The chromosomes are also involved in splitting the chromosome into a short arm and a long arm. As per the name, centromeres are not located exactly in the center, but is located at the end of the chromosomes. This organelle is also referred to as the chromosome arms, as they are present on either sides of the centromere. The main function of centromeres are the point of attachment of the kinetic core. The main function include the attachment of sister chromatids, and it is the site of attachment of spindle fiber. Second, centromeres help in the proper alignment and segregation of the chromosomes during the process of cell division in eukaryotic cells. In eukaryotes, the kinetic core is a proteinaceous multi-subunit assembly whose main function is to provide a site of attachment of spindle fibers to pull apart the sister chromatids at the time of cell division. Kinetic cores couple sister chromatids to dynamic matrotubule during congregation and anaphase, allowing their separation and partition to the daughter cells. Upon attachment, kinetic cores move chromosomes on the spindle, generating tension. In the absence of proper chromosome attachment and spindle tension, checkpoint mechanism prevent progression through meiosis, while the attachment of DNA to microtubule is clearly a fundamental role of kinetic cores. In most animal cells, the kinetic core is a disc-like compact forming on the side of every chromatid where spindle fibers would attach to. Thus, a human mitotic chromosome being comprised of two sister chromatids linked together via a centromere would have two kinetic cores situated in opposite direction at the centromere region. The kinetic core is comprised of an inner region and outer region. The inner kinetic core is tightly bound to the centromere DNA. The outer centicore is the one that interacts with the microtubule. Depending on species, the number of microtubules attached to the kinetic core varies. In humans, there are 15 microtubules attached to the kinetic core, unlike the outer kinetic core that forms and becomes functional only during mitosis and meiosis. The inner kinetic core persists throughout the cell cycle. Spindle fibers form a protein structure that divides the genetic material in a cell. The spindle is necessary to equally divide the chromosomes in a parental cells into two daughter cells during both types of nuclear division, mitosis and meiosis. During mitosis, the spindle fibers are called the mitotic spindle. Meanwhile, during meiosis, the spindle fibers are referred to as the meiotic spindle. At the beginning of nuclear division, two well-shaped protein tubes called centrioles position themselves at opposite ends of the cell forming cell pole. Long protein fibers called microtubules extend from the centrioles in all possible directions, forming what is called a spindle. Some of microtubules attach the poles to the chromosomes by connecting to protein complex called kinetic cores. The word somatic comes from the Greek word soma, which means body. In the human body, there are about 220 types of somatic cells. Somatic cells are adult stem cells. They are found pretty much everywhere in your body, as they are the cells that replace old ones, repair damage, and contribute to growth and development. During mitosis, a somatic cell duplicates its DNA and divides, creating two identical cells. One stays a stem cell and the other differentiates into the types of cells that is needed. Somatic cells are found in niches in different organs tissues. They need to be activated in order to start dividing. There are many different kinds of somatic cells in the human body because nearly every cell found inside and on the surface of the human body, with the exception of cells that become sperm and eggs, is a somatic cells.
Gametes or reproductive cells are produced in a special kind of cell division called meiosis. During this process, the normal number of the cell's chromosome is reduced by half. Each gamete ends up with a copy of one chromosome from each pair. Meiosis is a continuous process, but for convenience, it is usually examined as a series of separate events with two divisions. Inherited disorders can arise when chromosomes behave abnormal during meiosis. Chromosome disorder can be divided into two categories: abnormality in chromosome number and chromosome structure rearrangements. Cytokinesis is the physical process of cell division, which divides the cytoplasm of a parental cell into two daughter cells. It occurs concurrently with two types of nuclear division called mitosis and meiosis, which occur in animal cells. Mitosis and each of the two meiotic divisions result in two separate nuclei contained within a single cell. Cytokinesis performs an essential process to separate the cell in half and ensure that one nucleus ends up in each daughter cell. Cytokinesis starts during the nuclear division phase called anaphase and continues through telophase. A ring of protein filaments called the contractile ring forms around the equator of the cell just beneath the plasma membrane. The contractile ring shrinks at the equator of the cell, pinching the plasma membrane inward and forming what is called a cleavage furrow. Eventually, the contractile ring shrinks to the point that there are two separate cells each bound by its own plasma membrane. If cytokinesis did not occur during mitosis, the cytoplasm would not be divided and there would not be two identical daughter cells as a result. So, the cell would remain to be at rest, not being able to separate into two individual cells. Cardiokinesis is the step during cell division where the nucleus divided to form two daughter nuclei. It is the first step in M phase. It is usually followed by cytokinesis. In this process, the DNA condenses and the chromosomal material divides equally into two halves. In this video also, I will share with you the differences between cytokinesis and cardiokinesis. Cytokinesis is the process of division cytoplasm, while cardiokinesis is the process of division nucleus. The cytoplasm, cells, organelle, and the nuclei get divided and passed on to daughter cells equally, while cardiokinesis is the process where the nucleus divides into two daughter nuclei. Cytokinesis can occur with dependent on cardiokinesis, while cardiokinesis can occur with or without cytokinesis. During prophase 1 of meiosis, homolog chromosomes become connected in a process known as synapsis. While autosomes always undergo synapsis during meiosis, sex chromosomes often remain unpaired. While in synapsis, non-sister chromatids may break and recombine with their homolog partner. This region called chiasmata, where non-sister chromatids remain physically connected at this point of exchange. Chiasmata hold the homolog chromosomes together as a bivalent until anaphase 1. Chiasmata formation between non-sister chromatids can result in exchange of alleles. Chiasmata are essential for accurate meiosis. In fact, cells that fail to form chiasmata may not be able to segregate their chromosomes properly during anaphase, thereby producing an euploidy gametes with abnormal number of chromosomes. Crossing over is the swapping of genetic material that occur in the germline during the formation of egg and sperm cells, also known as meiosis. Pair chromosomes from each parent align so that similar DNA sequences from the pair chromosome cross over one another. Crossing over results in a shuffling of genetic material and is an important cause of the genetic variation seen among offspring. Crossing over occurs between prophase 1 and metaphase 1 and is the process where two homolog non-sister chromatids pair up with each other and exchange different segments of genetic material to form two recombinant chromosome sister chromatids.